So thank you very much for inviting me. We're actually not that far apart on campus, although I mostly hear about you guys when you make it on the news or National Geographic or all those cool places where I can not really hope to be quoted. So I'm going to talk to you about the work we've been doing in the area of human genetics, some of the interesting problems that come up, some of the current state of things, and hopefully, if I can do it in time, then we'll have time for a little bit of discussion at the end. So, human genetics this century, so when you look at the history of human genetics in the last 15 years or so, one of the big things that's been happening is that every year we keep getting more data. And if you quantify it, it's about 4 times more data each year than the year before. If you then compound that over a few years, you say, hey, 5 years, you get 1,000 times more data. 10 years, it's 1 million times more. And so that poses challenges in terms of computation. It also says that the kind of questions you can answer keep changing, and they often get a bit more interesting. Some of the questions that were hard when you look back to early in the century, like identifying a genetic variant that predicts a particular complex trait that, say, predicts whether you're going to get heart disease or diabetes, those are relatively commonplace now. And the hard parts of the process are actually kind of trying to interpret that genetic variation. We used to make the argument, you can use genetics to study any trait. Once you identify these variants, you'll know the trait biology. But it turns out that it's not that we can identify these variants at scale, it's actually very hard to connect them to biology. Most DNA polymorphisms are not so easy to connect in that way. And so the other thing that I'll try and touch upon is that as these data sets get bigger, the standards for how you share and use them change. And so in this slide here, I just list some of the studies where I've been involved. And number of genetic markers we measured, and you can see it started relatively small, maybe 20-odd markers, hundreds, thousands. Back in 2002, measuring 1,500 genetic markers in one set of samples was a big challenging thing and got you a paper in Nature. By 2005, with inflation and so on, that's now up to 1 million, and the cutoff on what's kind of an exciting technical advance keeps moving. But you can imagine that, for example, if you're in the context of studying human disease, if your study has hundreds of samples and a handful of markers, you're doing things like taking a shot in the dark. So you say, I'm interested in the genetics of obesity. I think it's all going to be about how you metabolize sugar or fat. And you pick a gene involved in that process, pick a few variants in that gene and measure just those. You do the analysis, publish the data, and that data is probably, pretty much all summarized in that paper. It's not so exciting to look at it again and again. Now, by the time you get to measure millions of variants, you can measure every gene. And so I could repeat my obesity study. I might focus on a few genes where I think I have something interesting to say. But someone else could say, hey, can I have a second look at your data? Because it'll tell me things about the other genes that you didn't get a chance to discuss in the paper. And so the standard around this time became, if you do an analysis, you make the data available so others can have a second look at it. They can combine it with their own data. And the challenge when you get to the current data sets, as you'll see, is that even though in theory we still have this standard that, hey, if you want a copy, I'll give you a copy of the data. In practice, it's actually not so easy to do because the data is so large that very few people could afford to keep a copy of it on disk. And I'll give you some examples in a bit. Okay, just to get this in context, this was a review of genetic association studies for complex disease by Joel Hirschhorn and others back in 2002. And the key quote is that they took 160 signals that had been studied 3 or more times, these connections between a particular genetic variant or gene and disease, and they said, hey, for 160 of these, I can find at least 3 papers. Let's see if the papers give a consistent answer. And they said, hey, only 6 of the 160 were consistently replicated. And so at that time, it was thought to be a very big challenge to even find these associations. And the reason turns out to be that we were very bad at making those guesses. We'd be studying obesity and say, let's study the sugar metabolizers. And that's not the right place to look. Here's, for example, a study of the Genetics of Obesity from 2009. There's a number of hits that are validated here in green. This is a study of about 20,000 people. What turns out to be interesting is that in these genes, and in these losses, at least the ones we understand well, for example, this one is a neuronal growth regulator 1. This one is interesting, SH2B1, because it's one that had been studied here in Michigan in the mouse. If you remove this gene from a mouse, you get a super obese mouse. You put the mRNA back in the mouse brain, you go back to a normal mouse. And so nearly all of these seem to be acting in the brain. And so the key thing for human obesity is not how you metabolize fat or sugar. It's about how your body decides how much to eat, or how hungry you should be, or how much to exercise. I'm going to give you just a quick tour of a different human disease, just to make a couple more points, and particularly ideas on how one might eventually start moving from these genetic signals to biology. So macular degeneration is a relatively common disease in the elderly. About 4% of individuals at age 75, eventually about 12% by the time you're at age 80. And it's a kind of blindness that's associated with the death of your central photoreceptors. So you go blind from your central vision, then spreading out. We helped organize a consortium where we put together data from around the world, from 26 different sites. And actually, one of the first steps in these kinds of analysis is to just make sure that the data makes sense. So you could imagine things like, you put all the samples together. In this case, we had about 35,000 samples, 17,000 cases, 17,000 controls. And we checked, hey, everybody that says they're a male, do they have a Y chromosome? And if they say they're a female, do they have two X chromosomes? And in this particular study, those checks didn't go so well. And it turned out to be for an interesting reason. It turned out to be, this is the fraction of missing genotypes on the Y chromosome, that particularly as people got older, many males had much of their Y chromosome missing. And I guess what was interesting is if you go back in literature, this is a paper in the American Journal of Human Genetics back in '95, and they looked at individual cells in blood, and they also saw that the fraction of cells where the Y was missing is increasing with age. And so I guess if you define being male as having a Y chromosome, you'd say that as you get older, you're not as much of a man as you used to be, right? Because you're losing that. But you go from being scared that, oh, this data is terrible, it's got all these problems, oh, it actually makes sense, it's just biology. And then you compare all the cases and controls. We had several million genetic variants across the genome, so here they're laid out. We do a comparison for each one between cases and controls, and you get these sort of signals, some very strong. For example, here the complement factor age, the strength of the signal is 10 to the minus 800, so extremely different frequency of those alleles between cases and controls. And if you browse, you see, for example, complement factor I, complement 9, complement 2, complement 3. There's in total about 30 signals, and 7 or 8 of these, in the case of macular degeneration, are in the complement pathway. And so without being a genius in biology or biochemistry, you'd say, hey, maybe there's something between the complement pathway and age-related macular degeneration. And that's one of the early observations from human genetic studies. But then you still have all the other 25 loci or so to try and figure out. Sometimes there's when you zoom in, zoom into a locus, there's a set of associated variants, set of genes, and sometimes there's a very natural story to tell. This particular set of variants is near the VEGF gene. This gene turns out to be the target of a drug that basically stops progression in age-related macular degeneration for about half of individuals. And so you'd probably look at this and say, hey, this is telling me that VEGF is important. It makes sense with what I know. But sometimes you zoom in, this is another locus, and here there was sort of quite a bit more difference of opinion about which gene is important. And because these variants are all non-coding, in this case they're all in the intron of this large SYN3 gene, it's kind of hard to decide, is it SYN3 that's important, is it TIM3, is it something else? they're probably regulating the expression or something else about how these genes function, but they're not directly changing the protein coding sequence. They turn out to be actually also relatively common. They each have a frequency of about 20%. In this case, we had a clue, and the clue was that in TIMP3, there's a rare Mendelian disorder called sorbus, where 10 different variants had been identified. 8 of the 10 added a new cysteine to the gene. And so we designed an array that let us test for any extra cysteines anywhere in the gene, and went back to our set of 35,000 individuals and checked what we saw there. What we saw is that in the macular degeneration cases, we saw 28 variants that were either an extra cysteine or had been implicated in this rare early onset disease in the cases, but only 1 in the control, so kind of a pretty strong split. And so we'd say, hey, this shows that if you damage how TIMP3, particularly by adding extra cysteines that are going to change how the protein folds, you're going to put yourself at risk of macular degeneration. And we would say that actually this points to the idea that once you identify these regions and you have this list of candidates nearby, you could look for really rare variants, either that cause big disruptions like folding in this case, or early stops in genes, and use those to kind of hone in not just it's a gene in this region, but which particular gene it is. And what we would love to be able to do is to eventually do this for every gene in the genome and say, let's compare cases and controls for any disease and say, is there an excess of stops or other large effect variants in one group or the other? If you zoom in, you can also see things like, hey, the carriers of these TIM3 defects are a little younger than the other cases, 65 versus 77 years old. But it's still not like a Mendelian disorder might look like. So in this case, in controls in blue the frequency of CF-age risk alleles. These are all the other AMD cases, and the AMD cases that also have that those TIM3 defects are kind of in between. ARMS2 is another very big AMD risk factor. And again, you see those TIM3 cases are in between. So TIM3 increases your risk by 30-fold, but it's not quite sufficient by itself. It still needs a bit of help from all the other risk loci and alleles. And so in this case, we look back at the story and say, these common variants, that it's something in this region. We couldn't quite figure out what it does. We tried to see if they changed expression and so on, but it's hard to do it in exactly the right cell type and the right time. But then by studying these rare defects, we would now claim it's probably because of TIM3. Okay, I'm going to switch gears and talk a little bit about what's possible with sequencing, how those studies have evolved. So how close are we to being able to apply sequencing to every gene in that scale to very large numbers of genomes? I'm going to tell you first about the 1,000 Genomes project, and then show you some more recent data and some things about the scale of those data sets. And so the 1,000 Genomes project was a project to survey human genetic variation around the world. We ended up studying about 2,500 individuals from 26 populations, and each of these individuals gave consent for their complete genetic data to be available on the web with no data access restrictions at all or anything like that. And so one of the ways to summarize it, which to you all will probably seem extremely obvious, is to just say, hey, how many variants are in each genome? If we look at, say, single nucleotide variants, about 3.8 million per genome, then short insertions and deletions, about 500,000, and then more complex variants, fewer and fewer. These are ALUs and line insertions, about 1,000, very large deletions, about 1,000, other kinds of copy number variants, about 150 per genome. And if you search hard enough, you'll find all sorts of odd, unusual things. In this case, for example, we found each genome had an average of 11 pieces of sequence that were inverted. This part of the slide shows what happens when you look by population, and they're kind of color-coded. And you see, hey, in Europeans, we have about just a little bit over 4 million variants per genome. East Asians, about 4 million variants per genome. South Asians, 4 million. These are individuals sampled in the Americas, and you see a range, and then individuals with recent African ancestry closer to 5 million. And so this makes perfect sense, as you all know. Modern humans originated in Africa, much more variation there. A subset of them moved to colonize the rest of the world. They didn't bring all the variation that was in Africa. They brought a subset of that variation, and so we see less variation in these genomes. And then these ones here in the Americas are often admixed. They have some native European and African components, and depending on the proportions which you mix them, you get a range of total numbers of variants. What's really kind of cool is that not just can you see these broad patterns, but there's now, for example, it's been shown that actually you can look at these genomes in more detail. For example, you could segment them into little blocks and say, how many differences do I see in the two haplotypes for each individual? And you can kind of say, date, what was the most recent common ancestor for that block, right? If you have very few differences, you'd say it's very recent. If you have tons of differences, you'd say it's very old. And if you take a whole genome and divide it into many thousands of pieces, you get an idea of, say, are there times when many blocks coalesced and you say, hey, maybe the population size was small then because I have lots of segments that date to then. Are there times when I see no blocks dating back to most recent common ancestors there? Maybe the population size was large. And so we did that for each individual in each population and generated these curves. And here's what they show. They show that among the individuals we studied, if you go back to about 150, 200,000 years ago, there's the same pattern of human population sizes. And so they all have a shared ancestry beyond that time. And then when you look, say, about, in this case, 25, 50,000 years ago, there's this big separation in two groups. These are the populations with recent African ancestry. These are all the others. And so we would claim, hey, this is the signature of the out of Africa bottleneck, when some individuals moved out of Africa to colonize the rest of the world. And then you see expansions. Eventually, they would reach, hopefully modern sizes. As it turns out, this particular method of segmenting genomes and doing one genome at a time is not very good for recent time spans. There's just very unlikely that there'll be coalescent events there. And so it's telling you more about what happens starting from a few thousand years ago. Now, another interesting thing when we looked at the 1,000 genomes data and compared populations, if you look at all variants, there's relative amount of variation colored by by population. More variation in Africa, less in the rest of the world. Things that change transcription, more in Africa. Synonymous variants, more in Africa. Non-synonymous, there's a lot more in Africa. Loss of function, things that truncate proteins, more in Africa. And so you say, hey, there's a pattern. But then you get to this end, and what's going on here? What are these two groups of variants that look so different? So this is variants that have been associated with common diseases by genome-wide association studies. And there it looks like, hey, there's more of those in a European genome. Why is that? Or these are variants that have been associated with, severe Mendelian disorders in this NIH database, ClinVar, more of those in European genomes. And so is this because Europeans have more genetic disease? And it's actually not because of that. It's actually just reflecting big biases in what human genetic studies so far have looked at. They've mostly looked at European individuals, and so we can annotate these disease variants in European genomes very well, but we can't see them. They're kind of in blind spots because we haven't studied them yet when they're in genomes of other ancestries. Okay, one of my favorite stories from the project was that when we started in 2008, we thought that doing a first pass look at the first couple hundred genomes would take about 70 or 80% of the sequencing capacity in the world at the time. Now it would be probably easy to do in the lab that we have on our campus, within a few weeks. But at that time, we were generating this data, and actually a big part of it was trying to figure out how to make sense of it. When you sequence a genome, you chop it up into little pieces, then you have to put them back together. We had three main analysis groups, one at the Broads, in Boston, one here in Michigan, one at the Sanger and the University of Cambridge. And what we decided was that we would each take our best crack at the analysis, and then we would compare results and see who did best, and say that would be the official result for the project. We would do a series of experiments to validate what we were claiming about each genome. And so we had different ways to measure error. So one was, for example, what was error at sites that when we validated, we knew were homozygous and should match the reference. And the Broad did best at those. But another one was to look at heterozygotes. And we had about 3% error rate at that time in those sites. And we would claim, hey, that's what you care about. It's heterozygote sites. That's the most interesting kind of place to look. But you could also look at homozygous sites that don't match the reference genome, and then the Sanger did best. And these are just three of the error metrics we had. And in fact, we were deadlocked for a long time because we would have arguments both about what was the right metric, or the Sanger guys would say, hey, we delivered our results in time. You guys were all months late. If it's a tie, we should take our results by default. And our funders were actually getting very frustrated. And at one of these calls, one of them said, you guys have three different analyses, and every base in the genome, you have, these three solutions. Why don't you vote? You say two of them agree, you go with that, then you go to the next base and vote again, and just go through every genome until you've scored it all. And I remember thinking, how could you let two wrong answers outvote the correct one? But since they were our major funder, I said, what an interesting idea. We'll check it out, and we'll report back. And so what was amazing was that it worked really, really well. So it was better at the homozygous reference sites than the Broad, at the heterozygous sites than Michigan, and at the non-reference sites than the Sanger. So it was better in every metric. And these were kind of, we thought, really state-of-the-art analysis methods that have been designed in very competitive ways because we all had the goal of having the official result for the project. And so you can take these good ideas too far, and in the final analysis for the final phase of the project, we ended up with 24 different initial analyses that we then merged in a complicated way to get to the final product. And this is all nice in some ways, but in the other hand, it meant that it took about from finishing the data generation here to having a finished product, in our final report out was over a year and a half. So it took quite a long time to go through all these analyses and square everything off. And so, in practice, when you're doing this for actual human disease studies, you probably have finite time, and you have to figure out how to balance this goal of doing all the different analyses and merging them versus getting an answer in reasonable time. Okay, so observations I hope I left you with so far. Human genetics gives us a general strategy for complex traits. These studies have been getting higher and higher in resolution. We can generate more data. We can look at more detail in any region. Common variants give us these clues about disease biology. And if we can get to combinations of rare variants, especially those that perhaps fully disable genes, we'll have much more definite clues. What are things that are going to be challenging over the next few years? One thing is if studies keep growing in size about fourfold every year, one big challenge for us is going to be, can we identify enough samples to study? Enough people that have, where we have phenotype and consent and genetic material. Another thing is that I think the standard where you say, hey, I'm going to collect these studies to study heart disease, and you collect yours to study obesity or schizophrenia or whatever, is going to start being different because we've measured all the genetic information. We're talking about such large sample sets that we probably should be able to do in the same set of samples many different analyses. And I think if we really get to this idea that we're going to replace, say, studies of knockout mice or knockout animals by searching the population for individuals with big natural defects in a gene and trying to get back in touch with them and studying them in detail, it's going to be more important to be able to stay in contact with study participants. And so one of the questions that we've tackled and what we've been thinking about is how can we get this dynamic phenotyping where we add extra detail as needed, and how can we remain engaged with research participants? So one of the things we've done is this study, which is probably in some ways the least appropriate place to do genetic research, but it's a Facebook app, and so people on Facebook search for genes for good. They fill questionnaires about their health. They fill out a consent form. We send them a spit kit. Then we share their data with anybody who's interested. And eventually, hopefully, there will be discoveries coming out. But at the moment, we already have participants in every state in the US. We have a set that's about 75% non-Hispanic whites, 25% other ethnic groups, which is relatively diverse, for example, compared to what we'd collect if we were collecting from the health system here in Ann Arbor. And and we have a way to stay in touch with them because we're in touch with them through Facebook. We do things like once we have their genetic information, we let them see summaries about ancestry. We let them compare phenotype answers with other participants. Here's a participant who told us about their alcohol use. And so we do things like summarize their weekly pattern for them. This particular participant drinks on Fridays and Saturdays. Not so unusual, I guess. And if you compare to all participants, they drink a little more, but a little less than other participants like them. So if you look at the data, it broadly makes sense. It's a relatively young sample, about 10 years younger than average US adults, but it replicates things that we expect. So if we look at BMI, as BMI increases, we see more diabetics, for example. The other study we've done along these lines is something called the Michigan Genomics Initiative, where everybody that comes in for surgery, we try to invite them to provide a DNA sample and consent to use their electronic health records in research. We spend a lot of time trying to make the consent form easy to read. And the reason is, so we got it down to about one page, one sheet of paper. And we've done some follow-up studies where we come back to people 3 or 4 weeks later and say, hey, do you know what you consented to? And with the traditional consents here at UFM, which are about 14 pages long, people will sign them. And then 4 weeks later, you say, hey, do you know what you signed up for? And they say, no. But here, with this one-page thing, they actually read them, and we get more people who know what they signed up for. And if we want to have the ability to, for example, go back and say, hey, you joined this study a year ago or a few years ago, now we want to know a little more about you, we're probably more likely to get that follow-up if people know that they actually joined in the first place. We tagged onto a little study that was recruiting more slowly and started recruiting at scale about 2012. When this slide was made, we were getting close to 30,000 enrolled participants. We now have over 35,000. And we've now collected the first round of genetic data on 20,000 and are starting to link it to electronic health records. And I'm going to show you just a couple of slides to just show that it actually works really nicely. These are the students and the team who handles recruitment and consent. So for example, here's a particular genetic variant. It's in factor V Leiden. And you test it here. Instead of being chromosomes, these are 1,600 different diseases that are relatively common in the Michigan electronic medical record system. They're grouped, here are cardiovascular diseases, and here, for example, are cancers. But this particular variant, you see very strong association with coagulation defects and also with deep vein thrombosis. But the nice thing is, in the exact same set of participants, and I have these 500,000 genetic variants, I can pick up different variants and see different signals pop up. And so here's one near TCF7L2, that one shows association with type 2 diabetes. And this one shows association with macular degeneration. That one shows breast cancer or dementia. And so it's kind of nice that in this set of 20,000 individuals, we can now answer, many, many different questions. And, and certainly more questions than between me and Chad and Sachin, who are the team who got this going, we could possibly go through. There's 1,600 different diagnoses in there. And so the goal here is that this is basically a research resource that different people could come up with different questions and ideas and explore. And you see interesting side things. It's hard to read, or impossible to read. But for example, in this breast cancer example, you'd see that this variant that's known to increase risk of breast cancer, and that shows that in our electronic medical records, also puts you at risk of esophageal cancer, or sometimes, for example, in that variant for deep vein thrombosis, we saw, strong association with coagulation defects. You can see related phenotypes. Sometimes these are connections that are known. Sometimes they're connections that are not completely expected. Okay, so I'm going to, for the last bit, and let's see how we're doing for time. Okay, I'm going to just give you a flavor of one of our large, this is actually our largest ongoing study. This is a study that involves many different groups in the US. They're sending their data to large-scale sequencing centers. The data then comes here. We have the role of kind of making sense of it all, processing it, sending it to a long-term archive at NIH. And we have a partner site in Seattle where instead of harmonizing the sequence data, they're kind of harmonizing the phenotypes and information about each sample, also then going to the NIH long-term repository. And so as of January this year, we had sequenced about 15,000 genomes. We had started sequencing them last summer. So, in this particular case, almost all of them passed quality checks. Then you see here 256, we thought there was not quite enough data as the spec. 128 had other quality problems. If you like math or do a lot of computer programming, you'll say, hey, 256 and 128, those are kind of special numbers. They're powers of 2. And you say, something wrong there. I thought there was something wrong, but it's just by coincidence. When I made this slide, those were the numbers. But we sequenced each genome about 37 times over. It includes about 99.4% of each one of these genomes. Contamination looks great. When I made this slide, I congratulated all the people who'd made the data and contributed samples. And we expect that by the end of this year, we'll have about 75,000 genomes sequenced. So this turns out to be 2 times 10 to the 15 bases of DNA sequence that we've generated and analyzed as of that point. And when you think about what's 2 times 10 to the 15, so one way is to say, hey, it's about the number of grains of sand in a small beach. But it turns out that there's actually a lot of controversy about the exact factor that you should use when converting from beach size to grains of sand. And so I could get from like a small beach to like the coastline of Maryland. And so I tried to get something that I could get a better handle on. And so it turned out that it's also about the same amount as the US corn production in 2014 if you measure it in kernels of corn instead of bushels. And so you could imagine that one of the things we're doing is we're saying, hey, let's tag each of these corn kernels by measuring something about it, something about how confident we are of those measurements. And you could say, we have different samples. Each of our samples is a collection of kernels, and we might say, this one has cancer or heart disease. Which of these little kernels is making that difference? And if you're more ambitious, you might say, well, why don't we say that DNA is, the genetic code that defines life? Maybe we can go from there to the muffin. We can reconstruct each person, right? And then we can do our analysis. But, and the catch in this turns out to be that you give the same sequence data to different people, and just like you have 2 cooks and the same recipe, there's some artistic license in the process. and they'll have slightly different results at the end. In this case, we saw that when we had the same sample sequenced in different centers, and we tried to harmonize the protocol exactly to say that we get the same answer, in fact, we were still getting up to about 3% differences for the same genome sequenced at different sites. When we get all the data to be processed in one site, basically all the centers generate the raw data, then it comes here, we harmonize it. We still have differences, but we usually remove more than half of the differences, and sometimes substantially more. I guess you can, if you're willing to not try and call every base in the genome, then you can get this even a little lower. Okay, in the analysis we've done as of December, we had about 10,000 samples, and this resulted in about 140 million genetic variants at single nucleotides and about 10 million insertions and deletions. So something like 1 in every 20 bases in the genome. We think by the end of the year we'll have 1 in every 6 bases or so. And if you look, and I guess probably no surprise to most of you, most of these variants are singletons. They're present in a single individual. Out of those 140 million, about 60 million are present in a single person. Only a small number are present in 10% of chromosomes. If you compare to, say, things that have been previously described, all these singletons are generally new, about 10% previously described, and almost all the common things had been seen previously. And as a first approximation, you could claim that that 0.6% is not previously described, or probably errors in our data. When we have many nuclear families, parents and offspring, an average child has 5,700 variants that are not seen in any other family, and about 1.3% of those, so about 60, are not present in the parents. An average child has about 300 short insertions and deletions not present in any other family, and about 1.7%, so about 5 or so of those are not present in the parents. Other kinds of interesting things one can check on already. You take all the variants, 40% are singletons. You take, say, the ones that change protein sequences. There's about 1 million of those. 50% are singletons. The ones that, say, add a stop codon, 37,000, 60% singletons. This makes perfect sense when you think about selection, right? So these things are probably not good for you. Nature doesn't allow them to become common, higher fractions of singletons. If you look at little insertions and deletions, 10 million, 40% singletons. The ones that are, say, multiples of 3 encoding sequence, 15,000, 50% singletons. So you could say, hey, these ones are more or less like an average SNP. These are more or less like a non-synonymous variant. And then the frame shifts, 30,000, more or less the same as the stops. And one interesting point is that, one is that we have about 2 of these and another almost 2 of those per gene. So if you think there's about 20,000 genes, almost 20,000, we have about 70,000 variants that are either stops or in frame. So we have 3 to 4 stops per gene on average, and about half of those are from the SNPs and half of those are from the frame shifts, even though frame shifts are only overall a much smaller portion of the data set. They're about half of these really interesting variants. One of the ways we check quality of the data is to say, hey, how many SNPs in each sample, how many indels. It's mostly driven by population genetics, so very, very strong correlation. Here's a little sample that looks odd. And so initially we thought, hey, this is a sample that failed our quality control filters. Somehow it got through, but it shouldn't have. It turns out this is actually an analysis error. This sample accidentally was sequenced instead of about 30x depth, about a few hundred x, and it just, the analysis tools we had were not prepared to deal with that. They had a lot of underflows and they started giving crazy results. So, something that we to fix. Now, this data set sits on about 4 petabytes of disk. You know, in theory, we're going to deposit at NIH. Anybody that wants a copy could go to NIH and say, hey, can I have a copy? And they'll say, great, you can start downloading it, and then you can either, you know, order a couple thousand hard disks or pay Google $1 million and make your own copy, right? But that's not so practical for many cases. And so we've tried to think about ways to make it easy to ask questions of the data without having to get a full copy. So we're putting together a variant browser where you can do things like pick any gene. Here's complement factor H, one of those macular degeneration genes, and eventually zoom into any particular variant, see stuff about it. This to say that, you know, when we built this based on an existing variant browser that's designed for exome data, and this is complement factor H there, and what you can see is, whereas before we had this really even pattern of coverage, which is current whole genome sequencing technology, when it was sequenced before, the coverage was actually quite uneven because it was using exome capture protocols. One of the things we're trying to put together is this idea that once you identify an interesting variant and would like to know something about it, we'll have a little click box. You say a little bit what you'd like to do, and then whoever owns that sample, whoever interacted with that participant, gets a request saying, hey, someone said they'd like to ask these questions about that participant. Is that possible? And you can then say, it's possible, this is what you need to do. Or in this case, hey, we're not in contact with the participant anymore. It's not allowed in the consent, can't be done. We also are trying to make so that you can do those kinds of analysis that I was showing for the macular degeneration data, those kinds of plots where you test association in a region. So they can happen basically within a browser. You don't actually, you pick the phenotype or you pick the region. You can move the mouse over, see details about each of these signals, but you don't have to download a copy of the data and manipulate it. And to give you just a last touch, you know, so I sit in a biostatistics department, so a lot of the things that I do is actually kind of work on the computational methods that underlie these analyses. In the last few years, one of the things that we've worked on is this idea of genotype imputation. So in humans, there's often inexpensive ways to measure a few markers, and then there's a set of individuals that have been measured in more detail at many markers. And we like to use this idea that we can find haplotypes that match, you know, so this one has A, and A. It matches this A, and A. This one, there's no perfect match, but maybe I can match it with a piece of this one, a piece of that. And I can then extrapolate and say, hey, maybe now I can fill in all the missing bits of data. And, you know, and we could argue whether this is a good or a bad idea. You know, the intuition that's a good idea is we're all related. If we share a stretch of chromosome, things within that stretch probably match as well. In fact, it's also an empirical question. You can design experiments to test whether it works. In humans, it turns out that it works incredibly well, and it works better and better as this reference panel either gets larger or gets to be made up of closer relatives to you. If it's, say, from the same part of the world, it's better than if it's from a distant part of the world. One challenge is that as these reference panels get larger, this gets to be more challenging. So in 2007, when we first did this, the best available reference had about 60 individuals, and analyzing a genome took about 14 minutes. 2009, there were still about 60 samples, but measured more carefully, 40 minutes. And it actually scales quadratically with sample size. And so you can see by the time we get to the 2012 data, 7,800 minutes, that starts to be real time. That's about probably 7 days or something like that. And if we were going to try and scale it to our current best set of reference samples, 33,000, it would take a very, very long time. The cool thing is that it's been possible to keep inventing new methods, new ways to answer this question. And so you can actually say, hey, if we were stuck with the 2007 software, it would take those 7,800 minutes. But in 2010, we had some new ideas about how to do it. Got down to 512, then 24, and now currently less than 1. And the other interesting thing, although we don't have time to go look at all the details, is that in part it's because we have more efficient solutions. But in part it's because these solutions are actually quite different. So the 2007 methods were really designed to deal with a lot of uncertainty, very little data. These newest methods assume that you have very large references and really hone in on long shared stretches. And so they work just as well as the 2007 methods, provided you have a large set of samples in your reference. They're actually very bad if you have a very uncertain, very small reference. But by taking advantage of the fact that the data is changing, we can find much better, efficient ways to compute on it. And in this case, we've actually, for example, it's now so efficient that we've put together this little imputation server where people doing human genetics can upload a sample that's been analyzed with an array. It gets compared to a panel of about 33,000 haplotypes from Europe, and then they get back a filled-in genome. And in the last year or so, it's been used to analyze over 2 million genomes. And just like I said, there's this bias in current human genetic studies. A lot more studies have been in Europe. You see that here, more in Northern Europe, then less good coverage in Central Europe, and the least good as you go to Southern Europe and Turkey. And if you zoomed in to show the rest of the world, current sequenced humans and haplotypes are not very good to get coverage as you get further and further out. Okay, the secret of coming, giving these talks and having all the pictures done and so on is having lots of really good students and postdocs that have worked with you over time. And so I think that was it for me. Thank you. I see. So how did we sequence these samples for these genomes? Is that what you mean? Yeah. So it's currently the standard is basically whole genome shotgun, and so so you take a sample of DNA, you shear it into little fragments of a few hundred bases, and then we usually sequence, about 100 to 150 bases at a time. Those reads are done in kind of in parallel. So actually, you shear a genome, get all those little few hundred base fragments, put them on a glass slide, and then you engineer a reaction where you sequence one base at a time in that glass slide and take a picture, shine light on it to see what you're sequencing. And so it looks like fuzz on a TV. Imagine like if you had an old TV and you tune it to nothing and you see little dots flashing on and off. And so basically you hone in on a dot and watch what happens over time, and that's the sequence that's flashing by. That method is really inexpensive, so you can sequence hundreds of millions of things on a single glass slide in parallel. It also has a relatively high error rate. So the error rate's probably about 1% per base. A human genome probably differs from another human genome about 1 every 1,000 bases. So the reason we end up sequencing 30 times over or so is because we need to get past that error, and we also need to sample the 2 chromosomes in each person. So I don't know if that answers your question, but that's my attempt. Right. So, the answer is, it is a really good question, and we've sometimes done a little bit of it, but actually, each of these fields and mushrooms, so a few years ago we did do a little bit in expression, then it's just, hey, there's so much to do there, we can't keep up, because it's about, do you measure the right tissue, and so one big difference, at least in humans, between looking at expression and looking at the genome is that genetic variation seems to give you kind of much more precise signals. So if I say compare gene expression between, say, people with psoriatic skin and normal skin, something we've done, you see thousands of genes pop up. And it's actually hard to decide which of those genes are kind of the cause and which of those are the effect. We like to believe that most of the time DNA precedes disease. And so if I take, say, individuals with psoriasis and individuals without, and I compare them, usually I get a much more focused set of genes. But yeah, they're both good experiments to do. If you look around the university, there's people doing both kinds of experiments, for sure. Yeah.
There was an interesting paper a couple of years ago, I think, that showed that if you could take anonymous genetic data and actually likely identify, make it de-anonymous, you could figure out who the person was. And I was just curious, thinking about there are genetic privacy issues, how they're driven to this process.
Yeah, so I mean, so we tell participants there's a risk this will happen. So the catch is basically that you can write in the data use agreement that you shouldn't try and identify participants, but it turns out that if you're willing to spend the time to do it, genetic information there's a lot of data there, and it's pretty informative, right? You could imagine that even if I just tell you in these 10,000 people, these are all the genetic variants I saw, if you say, you sequence another person, you say, hey, I want to find out if they're in your data set, you know that that person will have 6,000 variants that are unique to them. Get those 6,000, say if a large fraction of them are in your data, you probably have sequenced that person or many close relatives. And so you can answer questions like that. If you take this to extremes, you can sometimes actually answer questions about the phenotype of the person. Are they sick with this disease or that? And so at the moment it's actually a big question in human genetics, because these are things that were not so clear 5 or 6 years ago. And so probably most of the people that we're studying at the moment were not worried about these potential risks. And so there's a balance there between, you think this is a serious enough risk, it's real, that you should basically not share your data or results, or do you say, this is actually, even though it's real, it's more of a theoretical thing that's unlikely to cause harm to anybody. I think there's better good for science for sharing the data, and different people have different balances there.
I'm wondering, getting back to your study of the genetic information that's associated with the occurrence of particular disease symptoms.
Yeah.
Are you ever able to say, conclude from a particular analysis that a particular symptom has a certain percentage of a genetic contribution and the rest is environmental? The rest
I
is
see.
non-genetic?
Yeah, it's a question that people like to ask. And actually, so if you go into genetic studies, a few years ago, there was this big controversy about, what people call missing heritability. And the basic idea is that if you say do a twin study and say, in a twin study, it looks like height is 80% genetic. But then you say, hey, I measure a couple million genetic variants. Say, how much of height can I explain with those? And usually you end up coming up with something that's quite a bit short of that 80%, perhaps maybe for height about 30 to 40%. Why is that disconnect? And you can make lots of different hypotheses. You could say, hey, maybe in the twin studies you're capturing some of the environment as well as genetics.